Caskey from Baylor is going to tell us about some of those medications and the future.
Thank you. I'll make comments in 4 areas that I consider relevant to the development of therapeutics for Alzheimer's. These include a historic accounting of the coronary artery disease, intervention studies for another chronic onset adult disease. 2 is a review of the current Alzheimer's targets and the clinical status. And 3, I want to discuss a new therapy strategy. And finally, I want to discuss new genetic technology, which I think promises for new target developments and biomarker development. So let's start with coronary artery disease. In 1987, the FDA approved the first statin on the basis of cholesterol lowering, a biomarker. 7 years later, the Scandinavian study indicated that statins lowered the incidence of coronary events. It took 5.5 years to replicate this study in a U.S. population. Statins act by inhibiting the cholesterol biosynthesis. Genetic discoveries since that time have identified additional targets in the pathway: GI uptake of lipids, recycling, dietary load, and the most recent one, of course, is the LDL enhancer, PCSK9. All of these led to new FDA-approved drugs around the theme of cholesterol regulation. It was a genetic and pharmaceutical success story illustrating the time required for this success. There really is no quick fix on chronic onset adult diseases. We've got to keep that in mind. So let's talk about Alzheimer's. Genetic family studies have established that mutations in APP and the processing enzymes, the presenilins, are important in the development of the heritable forms of Alzheimer's. The fragment peptide, the 1 to 42, has been implemented as a toxic seeding fragment, and a second protein, tau, which was referred to, and its phosphorylated state have also been implemented in the pathology. The story for tau has gotten more interesting recently when it was discovered that tau mutations can also provoke the clinical features of Alzheimer's independent of APP. So there's a pathway that's developing around this idea. One other element which was mentioned earlier is the finding of the 4 to 6-fold increase with ApoE4 and the protection with ApoE2, variants in same structure. And the feeling is that this probably is involved in the A-beta clearance mechanism. So where do we stand on drug development? Inhibitors to presenilin 1 and 2 have had on-target toxicities, which occur by inhibiting of the Notch pathway. More medicinal chemistry, of course, will be needed to try to sort out this toxicity of an on-target effect before we can take this into chronic, take it into chronic therapy. So having been in Merck for many years, I've seen this before. The medicinal chemists can work miracles. Give them a chance to revise the opportunities to go after these 2 targets. The regulation of ApoE4 and 2, I think, potentially are worthwhile, but not a lot of specific targeting for regulation of these 2 alleles has been put forward. And then, of course, the strategy of using the monoclonal antibodies to trap and remove amyloid is rampant in the industry right now. Only Biogen has announced success in this area at this point, and they base that on PET scanning, resolution on PET scanning, and cognitive testing. They've made a big gamble by moving directly from phase 1 into phase 3 with a very substantial financial commitment on this gamble. I wish them well. The NIH studies utilize genetic risk identification for the candidates of their studies. And then Biogen used a different selection. They used PET scanning and cognition. So what are the issues that come up then, given this sort of mixed result on the antibody strategy? The trial issues are, who are you going to put on the trial? When are you going to put them on the trial? What's the dosage of the therapeutic you're going to deal with? And what is the agent that you're going to use? These are big uncertainties requiring biomarkers for the rapid decision-making for adaptive trial changes. I cannot emphasize enough how much more work we need in trying to get these signals of responsiveness on the targets that we believe are important. I continue to be very optimistic about this strategy. And I want to remind you of the HER2 story for monoclonal antibodies. What was not, I think, in the general literature is there were 1,000 monoclonal antibodies which were worked down the pipeline, and one was selected for clinical trial. We now know that the differences in the sites of binding for monoclonal antibodies vary the response to these signals. And then we get amplification in the case of the cancer area by moving to monoclonal antibody conjugates. You carry a drug package together with the monoclonal. So these are, I'm optimistic about it, but we have a substantial challenge. And just remember the 7-year trial period and the 5.5 trial period for coronary artery disease. Now I want to shift now a little bit to immune protection that relates to both Parkinson's disease and Alzheimer's disease. Just as we now know that self-immune recognition of cancer cells modifies the output and protection of individuals for cancer, there is the possibility that we may have the same kind of events going on with Alzheimer's. Now, the case is made really well in cancer by the approval of the new PD-1 inhibitors. I don't think anyone denies the fact that immune modulation, self-monitoring of immune response is going to be important. We know that both in Alzheimer's patients and in Parkinson's patients, that a certain fraction of them will carry elevated levels of antibodies to the target proteins, APP and synuclein. The question is this protective or complicating? And I don't think we have an answer to that yet. If you look at the trials that have been used for monoclonal antibodies in animal model systems, you'll find reports in both areas, protected, complicated, because it created inflammatory response. So it's an important thing for us to keep in mind that we think we understand a pathway, but there could be other elements. I remind you of the fact that cholesterol lowering hasn't stopped myocardial infarcts. That's a second process. That's an inflammatory process at the area of the vulnerable plaque. And therapeutics for that will be definitely targeted differently than just cholesterol lowering. So I've made my case that we need to do more work on the immunity reaction of these foreign proteins that are being deposited both in Parkinson's disease and in the case of Alzheimer's. Now, what about new targets? Geneticists in the past, and I put myself in the past, we just think it was wonderful to find a family with a lot of disease and study that family by mapping out the locus and making the discovery. And many of us in the audience here have done that. We don't do that today. We do individual patient studies because we've got a combination of marvelous new technologies in genome sequencing and imaging technology and metabolomics that make it possible for us to study a single individual with a disease and to come up with the conclusion this is a gene-disease causative relationship. And that's already been mentioned as the strategy by which we can move rather quickly on this. Now, what are the challenges of that strategy for the N of 1 study for new markers? And they are, one, the confirmation and the replication. I don't think that this can be done, that we can attack this by traditional scientific publication. It's just too slow, and there's too much gatekeeping on the admission of N of 1 patients into the literature. So how are we going to do this? I think it's going to be done by groups that that have the ability to master the technologies, and I'll name the key ones that I think are important, and that is gene sequencing, the imaging technology, and metabolomics. You may add to that list. But you have to have a single site competency to be able to carry out those studies with great precision. And then you aggregate the findings of these technologies to be able to draw conclusions on disease-gene relationship. So it's the new technologies, I think, that will give us the new biomarkers. Now, how do we replicate or confirm the observation? It will be done in the cloud. Other investigators with similar findings, before publication, will be able to access data of the cloud and to say, aha, I have a patient that is very much like this with this same disease gene association. That is the future. I'm probably overselling it as a geneticist, but I think this is the truth. So thank you very much for the opportunity to make some comments.
That's great. So thanks to the whole panel.